So then I was talking about this one. 70s ribosomes still can be seen in mitochondria and chloroplast. Actually, there is a small story behind this one, okay? But I'm not sure whether it is correct or wrong, but we believe that. Okay, then a cell wall. Cell wall. In cell wall, we can usually find peptidoglycan in bacteria. Peptidoglycan in bacteria. And we call this as also murine. Murine. I think last week I gave this is did I give this word murine?
No, sir.
Oh, okay. Then I want to check the spelling. I think I gave the word. I can remember I checked the spelling. Murie, Murine. Okay, N-E, Murine. Murine. Okay. This is the murine and peptidoglycan. We can see in the prokaryotic cell, but only write the word peptidoglycan for the paper. Okay. If you are asked to write two, then you can write peptidoglycan and murine. But if you are asked to write one, write only peptidoglycan. Understood?
Yes, sir.
Okay. Then in the eukaryotic cell. In plant cell, we have cellulose. Plant cell wall, we have cellulose, and fungi cell wall, we have chitin. We have chitin. Plant cellulose, fungi chitin. Okay. Then DNA replication. This is occur in the cytoplasm, occur in the nucleus because in the prokaryotic cell there is no nucleus, no. So DNA replication is happen in the cytoplasm. Cytoplasm is the area. It's actually jelly-like area. Okay, jelly-like area which all the organelles are embedded. Or we can see all the organelles are in this cytoplasm. Understood? In this bacteria cell, okay, or in the prokaryotic cell, there is no nucleus, no. So DNA replication can be seen in this cytoplasm. Cytoplasm in this blue colored area. Blue colored area here. It is yellow colored area. Yellow color. Then this is green colored area, and here a dark brown color area. Understood?
Yes, sir.
DNA replication mean actually making a DNA, making two DNA from a single DNA or one DNA, making another DNA from a existing DNA. That is called as the DNA replication. This is a very complicated process. Okay, we don't have to learn about DNA replication under our syllabus, and actually this is quite complicated process. Okay. Then next we have example. An example bacteria and archaea. There are a lot of archaea. There are a lot of archaeas, but in our syllabus we met only methanogens. Methanogens. These methanogens are met in biogas production because they are responsible of producing methane. They are responsible of producing methane. Okay. Then here examples are plants, animal, fungi, and protist. Plants, animal, fungi, and protist. When you talk about the evaluation, the I told you prokaryotic cell are the first organism or the first cell type which was origin for the first time. That means before 3.5 billion years ago. Before 3.5 billion years ago. Okay, not million, okay, billions years ago. Then these are evolutionary very recent years. That mean before 2 billion years. Reason mean actually reason mean not yesterday or last year. Okay. When you consider or when you compare about these two, this is quite recent. Okay. This is quite recent and this is quite what's called a historical, just like that. Okay. Then there's a special feature. There's a special feature which can only be seen in prokaryotic cells. That's the nitrogen fixation. Nitrogen fixation. This nitrogen fixation can be only seen in bacteria. In eukaryotic cells, you can't see any nitrogen fixation. Okay, here right. Some bacteria are capable. Some bacteria are capable. Some bacteria are capable. As an example, cyanobacteria, rhizobium lipase. Some bacteria. Within brackets, cyanobacteria, cyanobacteria. Although we have to talk about this one later. Not later, in the next lesson, okay? 4.2 lesson. In rhizobium. Cyanobacteria, rhizobium.
Done, sir.
Okay. There are a lot of bacteria, but for here, those cyano. Okay. Then presence of DNA, we talked about this one. Both of the cells have DNA. Chromosomes shape, we talked about that one also.
Sir,
Then, yes.
so we should write some bacteria, so cyanobacteria and rhizobium in eukaryotic cell part, right, sir?
No, 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 no. In prokaryotic cells, those are
In
bacteria,
eukaryotic,
no? Sorry?
yeah, sir.
Those
In
are
eukaryotic cell,
no, no.
is it
That's
blank?
why I told you. There are no any nitrogen fixation in eukaryotic cells.
Okay, sir. Done, sir.
Okay. Here, presence of DNA, chromosome shape, that is okay. Then let's move to the main cellular organelles and their functions. Main cellular organelles and their functions. You can see most of the cellular organelles cannot be seen in prokaryotic cell. Here ribosomes, cytoplasm is present, cytoskeleton, that is not too much important, and cell membrane. Cell wall is present, nucleoid, plasmid, flagella, pili, capsule. Okay. Here nucleus. The functions of nucleus is the main function of the nucleus is it is the it is just like a commanding officer. Okay, commanding officer. That means it controls all the cellular activities. It controls all the cellular activities. controls all the cellular activities. And within this nucleus, we can see DNA and gene expression and DNA replication site. What is mean by gene expression? Gene expression. Gene expression mean gene expression mean all the genes within ourselves is known as the gene expression. That means my gene expression and your gene expression is not similar. That means actually it is similar around more than 99% it is similar. No, more than 99, I cannot remember the exact percentage, but more than 99% is similar. And there is a small, there is a small deviation, not deviation, there is a small changes, okay? There is a small changes. The everything is regulated by the gene expression. That means your height, weight, and what is called your memory capacity, brain capacity, then your skin color, your hair color, your eye color. Most of the things are controlled by this gene expression. Okay, but in some cases, we can deviate the gene expression. Just like think like this. Uh, According to your gene expression, okay, according to your gene expression, uh, you must be overweighted. So you must be overweighted, but by doing some regular exercises, you can reduce the weight. Understood?
Yes, sir. Understood.
Not only in only some cases, okay? Only some cases, you can do like that kind of thing. Okay, DNA replication means uh, from the existing DNA, new DNAs are formed. New DNAs are formed from the existing DNA. And that's all about the nucleus. And we can see nucleus only in eukaryotic cell. Then mitochondria. Mitochondria. Mitochondria is considered as the power station of a cell. Power station of a cell. Okay. Power station means it can generate energy. It can generate energy. Actually, this energy mean it is a molecule. Okay. Energy mean it's a kind of molecule. That molecule is known as ATP. Adenosine triphosphate. Adenosine triphosphate. Okay. There are three phosphate group in adenosine. There are three phosphate group in adenosine. So what happened when one of this bond is broken, energy is released. Energy is released. So when this bond is broken, there are two phosphate now. Then this molecule is known as adenosine diphosphate. This is triphosphate, this is diphosphate. Okay, when adenosine triphosphate become diphosphate, energy released. Understood?
Yes, sir.
Okay. This energy production, this process is known as cellular respiration. This ATP production or the energy production is known as cellular respiration. Cellular respiration. I think it's better to write here energy production. Otherwise, you will confuse with what is meant by ATP production. Just write energy production. Energy production under this one. Energy production. Okay.
Yes, sir.
Okay. Then uh, ATP production. First of all, the glucose in our body, okay, first of all, glucose in our body is used for the energy production or used for the cellular respiration process. First of all, glucose is used, okay, in our body. When we eat carbohydrate, it produces glucose, no? As the final product, it produces glucose. Then this glucose is used for energy production. When glucose is over in the body, then as the second molecule, fat, fat, fat is used for energy production. Fat is used for energy production. That's the second molecule. Third molecule is that means when all the fat is vanished and there are no more fat and as the final molecule, the body is going to use or body use proteins. Body use proteins. This is the molecules. Sequence of molecules. Understood? First of all, use glucose, then after glucose is over, then turn to the fat. Then all the fat are vanish. Then try to use protein. This is the energy production or this is the sequence. The sequence. Understood?
Yes, sir. Understood.
Okay. Then that is the reason some of the doctors and some of the coaches, that means gym coaches and some of the dietitians tell uh, is that to exercise in the morning, okay, to exercise, do the exercise in morning, but don't eat anything. But don't eat anything. That means if you eat the last day at 7 p.m., just think someone has eaten at 7 p.m. and in the next morning, when it become next morning around 6 to 7 a.m., all the glucose are utilized for energy production. No? Then there are no more glucose in the blood. Then if this person is going to exercise, there are no more glucose in the blood and fat is used for energy production. Fat is used for the energy production. Then he or she can burn the fat. Understood?
Yes, sir.
Okay. Then we have mitochondria is finished. Then right in here, this double membrane molecule, not molecule, double membrane organelle. Double membrane organelle. Write that also. Double membrane organelle. Double membrane organelle.
Done, sir.
Okay. Then Golgi, no, endoplasmic reticulum, please called as ER. ER, okay, endoplasmic reticulum. I told you there are two types of endoplasmic reticulum. One is rough endoplasmic reticulum, and second one is smooth endoplasmic reticulum. Okay, smooth endoplasmic reticulum. This rough endoplasmic reticulum mean this has ribosome. This has ribosome. That is not in here, no? Okay. However, this ribosomes synthesis protein because actually the ribosomes can synthesis protein, and when this ribosome and the endoplasmic reticulum is combined with each other, they can produce protein, and they transport this protein. They synthesis and transport this protein. Then when there are no any ribosomes in the it is known as smooth endoplasmic reticulum, and it can synthesis lipid and transport, lipid and transport. Okay, then there are two types of what is called as ER. One is rough ER. Rough ER, it has ribosome. Then smooth ER, no ribosomes. Can you write this one? Rough ER, ribosomes. Smooth ER, no ribosomes.
So, does smooth ER transports water and other minerals?
Water, no, no, no. It cannot transport water. As I know, okay? As I know, it doesn't transport water.
Okay, sir.
However, in your syllabus, so in A-level biology syllabus, transport water is not mentioned on the endoplasmic reticulum.
So, can you tell some examples which transports by smooth ER?
That is what is called the lipid. Lipid.
So then rough ER. Okay, sir. Okay, I confused it.
Are you clear?
Yes,
Are
sir.
you
Now
okay?
clear.
Okay.
Done, sir. Finish writing.
Okay. Then there is another what's called that function by smooth endoplasmic reticulum. That is, it can synthesis what's called that carbohydrate also. Lipid and carbohydrate synthesis. Yes. Can you write this in here? Lipid and carbs. Carbs mean carbohydrate. Lipids and carb. This transport mean this can transport the synthesized lipid and carbohydrate. Okay.
Okay, sir. Done, sir.
Okay. Next one is Golgi apparatus. Modify and packages protein. Uh, this word is not correct. However, it produces lysosome. Okay, it produces lysosome, and this Golgi apparatus can produce what's called that glycoprotein and glycolipid. Glycoprotein and glycolipid. Yes, producing the glycoprotein and glycolipid is known as the glycosylation. Glycosylation is the process. Is the process, and put the arrow and write synthesis of synthesis of glycoprotein and glycolipid. 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 GLY. Glycolipid.
Done, sir.
Do you know the meaning of glycoprotein?
No, sir.
Glycoprotein means when carbohydrate combined with protein, it is known as glycoprotein. When carbohydrate is linked with lipid, it is known as glycolipid. That's all. Combining carbohydrate with protein is known as glycoprotein, and combining carbohydrate with lipid is known as glycolipid. That's all.
Okay, sir.
Okay, next we have lysosome. No? Lysosome. Main, uh, yes, digestion of waste, break down harmful substances, cellular cleanup, and right. This has stored. This has stored. This has stored. This has stored hydrolytic enzymes. Hydrolytic enzymes. Hydrolytic. Hydrolytic. Hydrolytic enzymes. Actually, that hydrolytic enzymes are responsible for digestion of waste and breakdown of harmful substances. Hydrolytic.
Okay, sir.
Okay. Then vacuole. Vacuole. Vacuole maintain the turgor pressure. Vacuole maintain the turgor pressure. Turgor pressure mean turgor pressure. Okay. This is the cell wall and this is the vacuole. Okay. When this vacuole or this cell sap is fully filled, it can exert a pressure for this vacuole wall. Okay, vacuole wall, and it can what expand. Then what happens when the this inner this inner organelle is going to expand? It can make a pressure to the this cell wall. Understood? It exerts a
Yes,
pressure to the cell wall. That is known as turgor pressure. That is known as turgor pressure. Have you seen some of the plants? Okay, actually not some of the plants. Most of the plants are get wilted when water is not there. Water is not there. What happened? The turgor pressure of the cells are reduced. That's why it is going to wilt. Wilt mean not wilting and not died. Okay, before that one, before that stage, it's get quite wilted. That is due to the reduction of turbo pressure. Understood? And it can store water and it can store some of the what's called the pigment. Can store some of the pigment also. Then next one, we have chloroplast. Chloroplast. The main function is the photosynthesis, and it contains chlorophyll, produce glucose and oxygen. Produce glucose and oxygen. Actually, this glucose and oxygen are the byproduct of photosynthesis. Byproduct of photosynthesis. Next one we have peroxisomes. Peroxisome. This does the detoxification process. Detoxification process. No need to go deeper about this peroxisome. Just remember detoxification or detoxifies. Detoxifies means when some harmful biomolecule or harmful enzyme or harmful foreign matter is entered to the cell. Okay, enter to the cell. These peroxisomes come forward and minimize or the minimize or the reduce or remove the toxicity of that substance. Understood? Peroxisomes are responsible for removing the toxicity of substances. That means the detoxification. That's mean the detoxification. Next one we have ribosome. Ribosome, the main function is protein synthesis. Protein synthesis, and it can translate mRNA into polypeptide chain. mRNA into polypeptide chain. That is not too much important for you, but protein synthesis, this is very important. Next one is the cytoplasm. Next one is the cytoplasm. These ribosomes are ribosomes can be seen in both cells. Okay, eukaryotic and prokaryotic cells. The cytoplasm also can be seen in both cells. Cytoplasm, this is a jelly-like medium, and all the chemical reactions are happen. Not all the chemicals reactions are happening in the cytoplasm, and this provides supports for organelles. That's why I told you organelles are embedded in cytoplasm, and this help to maintain the cell shape. Maintain the cell shape, and write another one. This is a jelly-like structure. This is a jelly-like structure. Jelly-like structure. Jelly-like structure. Finish.
That's it.
Okay, next one is cytoskeleton. Cytoskeleton is actually this one also not too much important for you. This provides structural support, cell shape, organelle transport, and cell division. Not too much important to keep a mark. Too much important, but just remember there is something called cytoskeleton. This also like that on centrioles, and actually the centrioles are responsible for cell division, cell division, cell division. Okay, cell membrane. Cell membrane means this is known as the plasma membrane too. Plasma membrane, right? Plasma membrane. Plasma membrane.
That's it.
Okay, this cell membrane are useful for. Actually, this is act as a gate. Okay, this act as a gate because this controls the entry and exit of the substances. This controls the exit and the entry of the some substances. Not some substances, all the substances. Okay. If there is any harmful substances, this doesn't allow to enter into the cell. Enter into the cell. And this maintain the cell integrity and cell communication. Cell communication. And next one is cell wall. Cell wall. We talked about this one. This provides structural support, protection, shape, and yes, protection and shape. Then chromosomes. Chromosomes. All the DNA and our gene expression is within this chromosome. Okay. In eukaryotic cell, there are chromosomes with histone protein. There are chromosomes with histone protein. And there is next one called nucleoid. Nucleoid means there is something or there is another structure within the nucleus. This is nucleus and nucleoid is within this nucleus. Within this nucleus. And this contains circular DNA, site of DNA replication and transportation. Okay, then plasmid, this kind of small circular DNA carry resistant gene genetic transfer, and that's why we can use this is in genetic engineering. We can produce enzymes, hormones by using genetic engineering. Okay, I will explain in later lessons about this one. Flagella, this only can be seen in prokaryotic cells. Prokaryotic cells, and actually some of the eukaryotic cells also, yeah, but most of the